Hello friends, in this session Strongyloides stercoralis. Strongyloides stercoralis is another intestinal nematode which lives in the small intestine of man. Introduction: it is a small nematode which inhabits the upper part of small intestine of man, and there is a property known as parthenogenesis which is observed in Strongyloides stercoralis. Parthenogenesis means that the female worm can reproduce on her own without the help of the male worm. This property is known as parthenogenesis and it is observed in Strongyloides stercoralis. Morphology of Strongyloides stercoralis: the adult worms, the males are not seen in the intestine, and hence it has been thought that the property of parthenogenesis occurs in Strongyloides stercoralis because the males are not found in the intestine. In parthenogenesis, the female reproduces on her own without the help of the male worm. The females live within the mucosal epithelium of the small intestine. They are about 2.5 millimeter long. They have 3 small lips in the mouth. The posterior end is pointed. The vulvar opening is at the junction of middle 1/3 and posterior 1/3. The gravid female uterus has ovoid eggs which are about 50 into 30 micrometers size, and this worm is ovoviviparous. What is ovoviviparous? Ovoviviparous means, first is oviparous. Oviparous means laying down eggs, and viviparous means laying down larvae, and ovoviviparous means laying down eggs which immediately hatch into larvae. So this property of ovoviviparous is observed in Strongyloides stercoralis. It lays down eggs in stool, but the eggs are immediately hatched into larvae. So it is known as ovoviviparous. This is an adult worm of Strongyloides stercoralis. The eggs of Strongyloides stercoralis are generally not found in the stool sample because they immediately hatched into larvae. But if at all they are found, they are 55 into 30 micrometers size. They have a thin egg shell, transparent and oval, and they immediately hatch larvae. The larvae hatched from the egg are the first stage larvae, that is Rabidiform larva. The Rabidiform larva, they hatch out from the eggs and they are about 200 into or to 250 micrometers long. They have a short mouth and a double bulb esophagus. They may convert to Filariform larvae in the intestine and they can lead to autoinfection. They may convert to Filariform larvae in the soil and then enter the new host to cause further infection. The Rabidiform larva of Strongyloides stercoralis, they resemble the Rabidiform larva of Ankylostoma duodenale. But there is some difference between the two, that the former has a smaller buccal cavity than the latter. Means the buccal cavity of Strongyloides stercoralis is smaller as compared to the Ankylostoma duodenale. Filariform larvae of Strongyloides stercoralis are the infective form. They enter the host through the skin of the host. They are about 600 micrometers long. They have a short mouth and a cylindrical esophagus. They are the infective form of this worm and they enter the body of a new host through the skin. Epidemiology of Strongyloides stercoralis: it is a cosmopolitan parasite found in different areas in warm environments preferably. Even sexual transmission of Strongyloides stercoralis has been observed. There are animal reservoirs of Strongyloides stercoralis. Transmission: there are 3 possibilities: indirect, direct and auto-infection. In indirect transmission, in the soil, the Rabidiform larva will convert to Filariform larva and the Filariform larva will develop into adults and several cycles of the worm will take place in the soil, and after several cycles it may enter a new host. So this kind of transmission is known as indirect transmission. Then comes direct transmission. In direct transmission, in the soil, the Rabidiform larva develop into Filariform larva and the Filariform larva don't develop into adults in the soil, but they enter a new host and lead to continuation of the cycle in the next host. This is called direct transmission. The third one is auto-infection. In auto-infection, the Rabidiform larva in the gut itself, they might convert to Filariform larva and lead to auto-infection in the host. So transmission of Strongyloides stercoralis, there are three possibilities: indirect transmission, direct transmission, and auto-infection. Now the same thing is described here in a diagrammatic manner with points mentioned at different places. There are three possibilities. In the adult worm, they are living in the intestine of the man. The adult worm will fertilize. Fertilization may say Rabidiform larva will be produced. The Rabidiform larva will convert to Filariform larva, and this Filariform larva can enter the same host again, which can lead to auto-infection. That is one possibility. The second possibility is the Rabidiform larva enter the soil, they convert to Filariform larva, and the Filariform larva, they enter a new host. That is direct transmission. And the third one is indirect transmission, in which the Filariform larva will convert to adults and several cycles of the worm will take place in the soil, and after several cycles the Filariform larva, they will enter the new host. So all these three transmission routes, indirect, direct, and auto infection, they are shown in this figure. The same thing is described here in the text manner. So parthenogenesis is a property that is observed in Strongyloides stercoralis. Rabidiform larva may convert to Filariform larva in the intestine and cause auto-infection. Rabidiform larva can convert to Filariform larva in the soil and enter a new host. And Rabidiform larva can convert to Filariform larva and then to adults in the soil, and several cycles complete before they enter a new host. These are three possible outcomes in the life cycle of Strongyloides stercoralis. How does it enter the host? Entering the host through the skin. From the skin, they go to the circulation. From the circulation, they go to the right heart. From the right heart to the lungs. The lungs, they enter the alveoli. Through the alveoli, they go to the trachea, then to the larynx, then to the epiglottis. And from the epiglottis, they are swallowed into the intestine. The larvae, they mature in the duodenum and jejunum and develop into adult worms, and adult worms will lead to fertilization and the cycle will repeat. What are the clinical features of Strongyloides stercoralis? The larva, they enter through the skin, and entering through the skin, they can lead to allergic reaction, they can lead to itching to the host, and this is known as ground itch. Then the larva, they can rather than entering the circulation and going to the heart and completing the cycle, they might just migrate in the skin, and they might not complete their cycle, but they might just wander in the skin, and such larvae, they are called larva migrans. They will keep moving in the skin here and there, but they might not enter the circulation. And those larvae which are moving very fast, they are known as larva currents. In larva currents, they can move as fast as 10 centimeters per hour. And more commonly, larva currents is found in the thighs. The larva, if they reach the lungs, they can lead to pneumonitis. And what are the effects produced by the adult worm? The adult worm can lead to diarrhea, which can have blood and mucus in the stool. So even this worm can lead to dysentery-like picture. Even Trichoris trichura can lead to dysentery-like picture, and even Strongyloides stercoralis can lead to dysentery-like picture in which blood and mucus are there in the stool. So clinical effects of Strongyloides stercoralis include the effects produced by larva and the effects produced by the adult worms. Here you can see a photograph in which larva migrans is seen. They will just wander in the skin and not enter the circulation. That is called larva migrans. This is a diagrammatic representation of the life cycle of Strongyloides stercoralis. The initial larva formed is the Rabbitiform larva, and it converts to Filariform larva, which is the infective form. So the infective form of Strongyloides stercoralis is the Filariform larva. What happens in the immunocompromised host? In the immunocompromised host, the infection can become more severe, and repeatedly auto-infection can occur, and because of that it can lead to hyperinfection syndrome in the immunocompromised patients. The worms may multiply very rapidly. They can disseminate to various organs of the body, and they may enter the lungs, and massive larval invasion of the lungs can lead to respiratory distress. Other organs can also be involved, and it can convert into life-threatening infection. So in immunocompromised patients, the infection becomes much more severe. In lab diagnosis, larva currents is a pathognomonic feature. It is a pathognomonic dermatitis. So, and stool sample can be seen. Stool sample can be taken in which wet films are seen. In the wet film, the Rabbitiform larva can be seen. Since it is ovoviviparous, larval forms will be seen in the stool sample in the microscope. Then concentration techniques can be used. Formal ether concentration technique and Bayerman technique can be used. Latent infection may multiple samples can be taken for screening purposes. And Bayerman funnel gauge technique is a classical technique which is used for the diagnosis of Strongyloides stercoralis. Duodenal aspiration can be taken. Charcoal culture can be done. ELISA, immunoblot and eosinophilia are other features. Now, what is Bayerman funnel gauge technique? In Bayerman funnel gauge technique, a funnel is taken and warm water is filled in the funnel, and 1 gram of stool is added to the warm water in the funnel, and it is allowed to settle down. As some time passes, the larval forms of Strongyloides stercoralis, they will settle down at the bottom of the funnel, and then the knob is opened up, and some portion, some water is collected from the bottom of the funnel in another container, and in that container, from that container, if you prepare a wet film and see in the microscope, then you can see the larva of Strongyloides stercoralis. So this method is known as Bayerman funnel gauge technique, in which warm water is used for concentrating the larva of Strongyloides stercoralis at the bottom of the funnel. In treatment, thiobendazole, albendazole and mebendazole, and in control and prevention, education, hygiene, sanitation and sewage disposal. With this, we end the session. Thank you.